Hello, this is Colleen Fitzwater from the Lobular Breast Cancer Alliance. Today, I'm speaking with one of the newest members of our Scientific Advisory Board, Dr. George Sflomos. Dr. Sflomos is a cancer research senior scientist in the School of Life Sciences at Swiss Federal Institute of Technology in Lausanne, Switzerland. He spent 10 post-PhD years in the area of basic and translational research in oncology, with a specific focus on solid tumor cancers. He now focuses on lobular breast cancer. George, welcome to the Lobular Breast Cancer Alliance's Scientific Advisory Board.
Thank you so much for the kind and warm welcome to the Lobular Breast Cancer Alliance family. I feel really privileged to be part of such an exceptional and diverse group of individuals. Personally, I strongly support the mission of raising awareness and promoting research for cancer, and especially for lobular breast cancer. It is really crucial that we work together to bring attention to this disease. Once again, thank you for making me feel so welcome.
Well, you are very welcome. So, George, I have a couple of questions for you today. And the first one is, can you tell me about your professional background?
Yes, sure. So my main areas of expertise are in vivo preclinical translational studies and experimental model development in oncology, combined with a solid knowledge of cellular and molecular biology techniques. I hold a bachelor in biochemistry and a master in biotechnology. And I received my doctoral studies degree from the medical school of the University of Ioannina, Greece, where I studied the fundamental role of key signaling pathways in cancer progression. After completing my PhD studies, I relocated to Lausanne, Switzerland, and I became a postdoctoral fellow at the Swiss Federal Institute of Technology, EPFL, in Lausanne. So I joined the breast cancer prevention and therapy laboratory led by Professor Catherine Brisken. So my goal was to develop experimental platforms that were better suited for target validation and drug discovery. Since joining EPFL, I have played a crucial role in developing better animal models for breast cancer research, focusing on the hormone-dependent subtype called estrogen receptor positive cancer. I'm currently working on estrogen receptor positive in vivo platform, focusing also on invasive lobular carcinoma.
Thank you. And why did you decide to focus your work specifically on lobular breast cancer? Why did you make that switch?
I would say mainly for two reasons. Firstly, as I mentioned, my studies focus on estrogen receptor positive tumors and the role on hormones in carcinogenesis, in breast carcinogenesis. Now, research has shown that the lobular breast cancer is highly hormone sensitive and expresses estrogen receptor. Yeah, the second reason is that on a personal note, when I was doing my master's in biotechnology, it sounded like I developed a real passion for a protein that is called epithelial cadherin. So it's present in epithelial cells. It is really fascinating how this protein plays such an important role in allowing epithelial cells to stick and interact together, and with the microenvironment also. So it's like a glue. Now, this particular protein is a hallmark of lobular breast tumors, as it is not really expressed, is not present in most of the lobular cells, and also is a hallmark of a few other cancers, including certain forms of gastric carcinoma. So I'm really fascinated about this cellular biology of these tumors.
Interesting. So I know that much of your research obviously is focused on developing and then studying also ILC models. This is something that LBCA advocacy is also hoping to help with, since we know these models for research are very scarce. So can you explain the importance of ILC models for research and how studying these models can impact those who have lobular breast cancer?
These models are very useful because they play a crucial role in facilitating the transition of novel therapies, novel experimental therapies from the preclinical laboratory models, including the ex vivo models, meaning the models that we perform outside of an animal organism on a dish in the laboratory, and also the in vivo testing, so meaning the experiments that we perform in vivo settings in different organisms, and they facilitate the transition to the human clinical trials. Now, I mean, there are many other reasons, as like studying new treatments in using these preclinical lobular models, the effect of these new treatments, if it's positive or negative impact, and the prediction in order to go to the next phase, to the clinical phase. The importance is also of these models. We can use it as to prevent lobular cancer, for example, to use as a models to study the prevention, how the lobular cancer spreads, how the lobular cancer becomes resistant to therapy. Of course, it's very important to assess the new treatments also and the mechanisms of resistance of the new drugs using these models specifically. And I would say one very important part is also to identify the mechanisms of lobular metastasis using these preclinical models. And the lobular, we know that has a peculiar metastatic pattern that we can now easily and modeling in vivo. And that's very useful.
So can you explain exactly what you're doing and how the models developed will be used?
So I'm really passionate about researching lobular carcinoma using different preclinical models. But the task of researching lobular carcinoma poses the significant challenge that as there are only few lobular models available, and there is a pressing requirement for additional ones. So, yeah, together with my colleagues at EPFL, I have developed innovative in vivo techniques that has been deemed a game changer for estrogen receptor breast cancer preclinical modeling. So we use this technique for lobular breast cancer, and this technique involves the grafting of patient cells directly into the mammary milk ducts of animal models, and is known as the intraductal model. So I saw that the tissue microenvironment is critical for the growth of the estrogen receptor positive breast cancer, lobular breast cancer. It is important to mention here that the lobular cancer cells that we study in vivo display similar growth, invasive behavior, responsive to treatment, and metastatic tendencies as in the real clinical setting. So that's very useful for a researcher. I mean, additionally, using this intraductal model, I developed the first lobular intraductal xenographs. And when I say xenograph, is when we take the patient's derived sample and we implant it in a different living organism in the laboratory, and in that way we found that the lobular cells are highly dependent on structural proteins that surround them, and that the lobular cells themselves, they secrete proteins and enzymes also that control this extracellular matrix, which potentially opens new therapeutic approach and possibilities. So currently I'm working on creating additional in vivo lobular models using either established lobular breast cancer cell lines or freshly dissected primary tumors derived from both primary or metastatic lobular carcinomas. The overall aim is to better model and study the lobular carcinogenesis and the mechanisms of lobular metastasis. I hope in general that this research will contribute to the development of new treatment strategies that can potentially in the future will be used in the clinic.
We're hopeful of that too. So George, are the models that you're talking about, are they hard to develop? And if so, why?
Yes, especially the in vivo models are more challenging to develop because they require a strong technical expertise. We perform surgeries to implant these lobular cancer cells in a living organism. They also require additional and more resources and infrastructure. We're lucky here in EPFL we have a very strong core facilities that are integral to the lobular research we perform here. They provide the centralized access to the technological resources and expertise, such as equipment, database, computational databases, materials, other resources that are very useful. I mean, then also we need to, when we develop these preclinical models, we need to constantly validate and optimize in order to be more useful and to be the right model, using the right model to answer the right scientific questions. Then using these models, because human material is implicated in our research, we need to have ethical and regulatory considerations. So this is also time consuming to develop all these protocols. But most importantly, we have to be really very critical how to obtain and develop the most clinical relevant models to answer our questions.
So is there someplace researchers in other parts of the world can go to access these ILC models to work with, or is that part of the challenge with ILC research?
Yes, so actually we recently reviewed all available experimental models for lobular research in a study published under the name of Atlas of Lobular Breast Cancer, where in this study, actually, we comprehensively present the key characteristics and peculiarities of the available published lobular models. So with this study, we suggest to reconsider some existing breast cancer models, which traditionally used as a non-lobular models, but possess some key lobular characteristics also. This atlas also can guide investigators who are considering implementing lobular models in their research programs. And this was a big worldwide collaboration, and the authors included, for example, the founding member of the LBCA, the advisor board, Dr. Steffi Osterreich and Dr. Adrian Lee, and guest editors for this special issue of the journal Cancers included the leading members of the European Lobular Breast Cancer Consortium, Dr. Patrick Dirksen from Netherlands, along with Dr. Christine Desmet from Belgium, and Dr. Anne Solomon from France. So now within the European Lobular Breast Cancer Consortium, in collaboration with groups that currently work with lobular models, we are currently developing a centralized database of lobular breast models, with the goal is to eventually offer a comprehensive evaluation of all lobular models and to provide these models that can be accessible for transfer to academic laboratories worldwide in a collaborative basis. Of course, upon acceptance by the owners with a signature of the MTA for mutual transfer agreement material, because some are involved also human material.
So George, you mentioned xenographs and you told us what they were. Can you tell me, how long does it take to develop xenographs, and is it expensive to develop?
First of all, it really depends on the experimental design and the scientific question. So a xenograph experiment typically takes weeks, months, or even a year, especially if we want to study mechanisms of metastasis, dormancy. We really keep the experiment running for a year. It is indeed very laborious and expensive, because it's 24 hours, 7 days. We have to really be in the lab. And for example, at the moment of a scheduled surgery, or if a patient passes away, we have a collaboration with Professor Christine Desmed, which is also a member of the European Lobular Consortium, and her team in Belgium, where we really receive material from patients that they have enrolled in a study, that they donated their body. So our team is immediately informed and has to plan an experiment, a xenograph experiment. And this is very essential to identify really the scientific interest and determine the optimal approach for addressing specific questions using these preclinical models. So for a clinical point of view, all these collaborations, there are several departments that are highly involved. And of course, there are key logistical procedures, ethical committees, approval, and this is all very laborious, and it's a teamwork.
Thank you. So I'm going to change the topic just a little bit away from the science and research that you were doing to the question of what drew you to the Lobular Breast Cancer Alliance and wanting to become a member of its scientific advisory board.
I'm really thankful actually to people who like to engage in meaningful dialogue. And personally, I met a plethora of them in the broader lobular community, and of course in the LBCA also. So I feel really privileged to have been included in such an exceptional group of individuals.
Well, again, we're glad to have you. I know that you're also involved with the ELBCC, the European Lobular Breast Cancer Consortium Lobster Pot. Why do you think it's important that organizations like ELBCC and LBCA collaborate?
Indeed. I'm actively involved in the European Lobular Breast Cancer Consortium, a consortium that started in 2018 with the intention of developing a platform to bring together discovery scientists, translational researchers, clinical experts, all together with the common mission to enhance the comprehension, detection, prognosis of invasive lobular carcinoma. This organization is extremely important in facilitating the translational of the discovery science into effective clinical implementation. This is the aim. So now, of course, we are aiming at closely collaborating with LBCA to establish a stronger lobular advocacy movement worldwide. This is very important because in this way only we can achieve our common goal, which is the improved understanding and diagnosis of and treatment of lobular internationally.
Thanks. And I've also seen you personally interact with lobular breast cancer patient advocates. Have you used patient advocates to help with your research in the past, or would you like to in the future?
I completely understand and agree with your statement. Actually, yeah, my passion for science was actually fueled by conversations with the community members and especially the patient advocates for lobular breast cancer. I just to mention some examples here, the fantastic collaboration and discussions, for example, with Siobhan Freeney from the Lobular Breast Cancer Ireland. Her advocacy voice really represents an outstanding patient voice in research and education for lobular, because it's helping other patients with the same disease and the community members. So also, of course, with other lobular advocates as the founder of the Lobular Breast Cancer Alliance, LBCA in the past. Also from the Dutch patient advocacy, Ryan Terver, or from the Norway lobular advocacy, Ton Lian, and Claire Turner from the UK. So these are all very important interactions and partnerships and they are really helping researchers, and they are really fuel the passion for working even harder for a common scope for fighting against this disease.
That's great. A lot of our friends and collaborators you just mentioned there. So why is it so important to work with these advocates?
So advocates, for me, they have the power to increase awareness for lobular breast cancer and educate the wider breast cancer community and the public. So I think that patients have the potential to serve as kind of mentors and provide the mutual aid. So for me, talking to the patient advocates became really my drive and motivation for science. By working together, only by working together, actually, we can identify the most pressing questions that need to be addressed. So, moreover, the research advocates can provide valuable support in shaping studies and clinical trials, for example, as well as communicating and interpreting research results to the communities, to the public. This is very important, and it's not trivial. It is really clear that their work is really vital to ensuring that patients receive the care and the attention they deserve, especially for lobular breast cancer, which is an understudied disease, and it's really important the role of advocacy in that disease.
George, thank you so much for taking the time to answer these questions today. I know I personally and the rest of the Lobular Breast Cancer Alliance community very much looks forward to your time on the Scientific Advisory Board. So thank you.
Pauline, thank you so much for the kind and again warm welcome to the Lobular Breast Cancer Alliance. Many greetings from Lausanne, from EPFL, and I look forward to working with you and the LBCA in all directions.